So here we are, concept 11.2. We said the first step in this whole cell communication process was reception. So 11.2 says reception is a signaling molecule binding to a receptor protein, causing it to change shape. We've already mentioned before how this receptor mechanism is very specific. It's very much like a lock and key. So the signal protein or the signal chemical that's floating around, it won't necessarily interact with every cell it comes across. It only will interact with the cells that have the right receptor molecule that fits that exact particular signal protein or ligand. So it's very specific. And a shape change in the receptor is often the first step in this whole transduction process. So when that signal molecule attaches to the receptor molecule within the cell membrane, it usually changes the shape of the protein. And that's usually step one in kicking off this whole process. Most signal receptors are in the plasma membrane. They are proteins. Almost everything we're going to be talking about here is a protein of some kind. And so we'll take a look at a model in just a moment. But most of these signal receptors are embedded in the plasma membrane of the cells. So while every cell may receive the signal molecule, only certain ones will react, and the ones that will react are the ones that have the specific receptors that fit that molecule. Just like an analogy would be like a radio station broadcasts its signal far and wide. But that doesn't mean every single radio within a 5-mile radius is picking up that station. You have to tune your radio dial to pick up a specific frequency, and then you'll pick up that particular radio station. That's very similar to the way this cell communication works. Not every single cell picks up the signal, only the ones that are tuned to that frequency are, in reality, only the ones that have the right receptor molecule on the outside. Fits together like a lock and a key. Think back to when we were talking about the enzymes and how they have a particular shape and they have a particular area for the activity, the active site, and how the ligand or the ligand fits in that one particular area into the catalytic site of the enzyme. And if you change the shape of the enzyme in any way, oftentimes the process doesn't work. So it's very much very specific, like a puzzle piece or a lock and key. We're going to take a look at three main types of these plasma membrane receptor molecules. So these are kind of like the docking ports or the lock where the key is going to be put into place. And I hate that the vocabulary can be very confusing, but we're going to step through them one at a time. So the first one's called G-protein coupled receptors. We'll find out why it's called G-protein coupled. The next one is called a receptor tyrosine kinase, and a kinase is a word for a particular type of molecule. We'll find out. And the last one is called an ion channel receptor. And we already know a channel is kind of like a pathway. We know that ions are charged particles, so those words we've kind of seen or heard before. One of the things to keep in mind is that the cell surface receptor molecules, these locks or these places on the cell surface where these signal molecules attach, if they don't work properly, oftentimes that's very much associated with diseases and other problems. So if you have cell receptor molecules that aren't working properly, or they are disrupted in some way, that could be indicative of a particular problem. It's either caused by a disease or that is because of a particular symptom of a disease. So a lot of research concentrates on these cell receptor molecules and the various chemicals, signal molecules that kind of interact with them. All right, here we go with some heavy vocabulary. Let's start with the largest group of these receptor molecules. And the largest group of these receptor molecules is called G-protein coupled receptors. And of course, we try to be lazy and shorten that to GPCRs, G-protein coupled receptors. And the reason that they are called G-protein coupled is because they are a plasma membrane receptor molecule, and they work with another protein called a G protein. So why is this second protein called a G protein? Well, it's called a G protein because it is fueled by a molecule called GDP and GTP. And that's very similar to some other high energy molecules we've already talked about, ADP and ATP. So the diphosphate and the triphosphate, the ATP was the adenosine triphosphate. So GDP and GTP are very similar. They're high energy molecules. They carry energy kind of like batteries that we've talked about before. Very similar to the ADP, ATP that we've already discussed. So we have this G protein coupled receptor, and it works in tandem with a G protein. And the G protein actually kind of acts like the on-off switch for this particular receptor. If GDP is bound to the G protein, then that protein is off. It's inactive. When it goes into the active state, you have the receptor molecule that receives the signal molecule, and then it binds to the G protein. GDP gets replaced by GTP, and that's kind of like turning the switch on, and that activates the G protein for action, so it can do whatever it needs to do, pass that signal on down the line. Before you shake your head and say, I didn't understand a word of that, just hang on just a second. We'll go over it again, and we'll look at a picture, and then we'll kind of talk about it while we look at the picture, because that helps quite a bit. So just in general, these G protein coupled receptors are the most common form of the cell surface receptors. They're very widespread. There's a wide number of them. There's a wide variety of types of them. They're very diverse, and they too are very much linked to early evolutionary development. You can find these GPCRs in pretty much most living creatures that use this particular variety of cell communication. So let's take a look at a picture, and then we'll go back through the words again, and it'll make more sense. So here we have kind of a model of what it looks like. So here we have the cell membrane, the lipid bilayer. This would be inside the cell. This would be outside the cell. So here, a portion of this big long curlicue, this is the protein. Remember, proteins take a variety of shapes. This one kind of has the alpha helix shape. But this particular part of the protein is the binding site, the receptor site. So let's say you have a signal molecule that's floating around in the external liquid of the cell, and it locks into this binding site right here. Well, that makes a change occur in the protein, and it changes the shape of the protein. And then down on this side, inside the cell, this is the portion right here that changes shape and that interacts with the G protein that we talked about just a moment ago. So this is a G protein coupled receptor. It only works in conjunction with this other G protein that we're about to take a look at. So let's kind of step through this process of the G protein coupled receptors. You can also find this on page 211 of your textbook. That's the exact same picture here. So here we have the cytoplasm of the cell. Here's that big protein, the G protein coupled receptor right here. And then inside the cell we have the inactive G protein. It's attached to GDP, but that's kind of like in the off switch mode, so it's inactive. And then there's the actual enzyme that we're going to have do some work. So this is step one. In step two, we have a signaling molecule that finds its way to the G protein coupled receptor, and it locks into its particular spot. That causes this protein to bind with the G protein. So see how it kind of changed shape right down here at the bottom? When that signal molecule locked in, it changed the shape of the protein. Now that allowed this to connect to the G protein, and the GDP gets phosphorylated to GTP, and suddenly that becomes activated. Then this G protein that's now activated, go down here to step 3, is released, and the activated form connects to the enzyme and causes the change to occur. You can see the signal molecule leaves its position once that has occurred, and then in step 4 we're kind of back to normal again. So in step 4, this protein receptor could be able to accept another signal molecule. The GDP has basically given up its energy. It became GDP back from GTP, and this is no longer active. It's back in the off position. So it's kind of a multi-step process. Here's the receptor. It gathers the signal. That causes a change, which allows these two to bind together. That causes a change which activates the G protein, giving it GTP from GDP. That goes along and then activates the enzyme, which then takes care of whatever response we're talking about. And then everything resets back to its initial off phase, and the whole process can start again. Here's a more realistic view of the G protein coupled receptor. Obviously, we're talking about proteins which can be very complex in shape, so kind of confusing of a picture to look at here. But this would be the plasma membrane that you see here. In here we have cholesterol. Remember, we have cholesterol molecules that are bound within the plasma membranes in various spaces. And then all the blue that you see here is the protein receptor, the G protein coupled receptor that we were talking about. Alright, the next category of receptor we're going to take a look at is called receptor tyrosine kinases. And before, again, we freak out, let's slow down. Let's look at each one of these words by themselves. We do shorten this to RTKs, receptor tyrosine kinases. Let's talk about what each one of those words means so that we understand and we don't get too confused. So we know what a receptor is. Receptor just means it's a protein that has a bonding or binding site where a signal molecule can come in. So that's cool. We know that one. Tyrosine is just an amino acid. It's one of the many amino acids that we've kind of talked about previously. So tyrosine is just a particular amino acid that we're going to be talking about or used in this process. And then this word is important, a kinase. What is a kinase? Well, a kinase is an enzyme that has a particular activity. Right down here it says a kinase is an enzyme that catalyzes the transfer of phosphate groups. And we've actually seen this quite a lot, where ADP becomes ATP. We have phosphates that are attached and released. Well, a kinase is the enzyme that does that. So when you see the word kinase, you need to think, okay, this is the enzyme that either attaches a phosphate or releases a phosphate, and it makes ATP out of ADP or vice versa. Or like in the previous example where we were talking about with the G protein, back up to where we were talking about with the G protein, where GDP becomes GTP. Well, it's a kinase that does that. So the word kinase is just a general term that means an enzyme that catalyzes the transfer of phosphate groups. So a receptor, tyrosine kinase. It's an enzyme of some kind. It works with the amino acid tyrosine. It either adds or subtracts phosphates, and it's part of one of these receptor molecules that attached to a signal molecule to start this whole signal transduction process. There are a few differences between the receptor tyrosine kinases and the G protein coupled ones that we were looking at. The receptor tyrosine kinases can actually trigger multiple signal transduction pathways. So they can take one signal molecule and basically then turn that into multiple actions within the cell. We'll take a look at that in just a moment. The GPCRs, the G protein coupled receptors, usually only trigger like a linear path, one particular signal transduction. So you're going to see in a moment how one signal molecule with the RTKs can create a whole bunch of changes, whereas with the G protein coupled ones, it usually one signal molecule creates one change, basically. Just like with all of these protein receptors, if you have abnormal function of the RTKs, oftentimes that's associated with major problems, including cancers and other diseases. We're going to find out, we'll look at a picture, that the RTKs, they exist as a monomer, so a single chain molecule, and then they actually combine into something called a dimer. Di means two. So two of them will actually join together to form this couple, this group. So if you're old like me, you used to watch a Saturday morning cartoon where there were superheroes, where there were Wonder Twins or Power Twins, and they could activate their powers by touching hands. This is kind of what's going on. These single monomers combine together to form dimers, and that's when they activate. These RTKs, we're going to find out, have multiple tails, basically. And after they form a dimer, after they join with their twin and activate, we're going to use ATP to add a phosphate group to each one of those little tails, and that's going to fully activate it. So they have multiple tails, which means they can do multiple strands of activity, basically. Each tail can then trigger off different relay proteins and different transduction pathways, so one signal can become many signals doing many different things very quickly. So let's take a look at a model. So here we have a model of the receptor tyrosine kinases. So here they are acting individually as monomers, and you can see they have all these little tyrosine tails kind of sticking off of them. This tan color is inside the cell, the blue color is outside the cell, and the darker line is the cell membrane. You can see they have a kind of a binding site, a receptor site. Here's the little signal molecule floating around out here. The signal molecule will attach to the receptor site. So let's move over here. When enough of the signaling molecules are present, they will attach to two of the monomers, and those two monomers will basically activate themselves by becoming what's called a dimer right here. After they form the dimer, then what's going to happen is they're going to use ATP molecules to fully activate all of these little tails. So they're going to phosphorylate the dimer. They're going to add a phosphorus to all these little tyrosine tails, and then that activates all these little tails for activity. And then there can be a wide variety of responses. You can have inactive proteins that can then attach to these little activated sites, and this one could go off and cause a particular change, and this one could go off and cause a different change. So you could have 2, 3, 4, 5 different pathways of activity going on, all starting off with this one single signal molecule. So this one set, or these are 2 examples of the signal molecule, can set off a whole chain of events because of this particular type of receptor. And again, this is called a receptor tyrosine kinase. Remember, a receptor, we know that what that means. Tyrosine is just the type of amino acid that it's dealing with, and a kinase is just an enzyme that either adds or subtracts phosphates. All right, the third and last one we're going to look at as far as receptor molecules is called a ligand-gated or a ligand-gated ion channel receptor, and this one is fairly simple. It simply acts like a gate, or when I think about it, I think about like the crossing arms that come down when a train is about to pass, and when the arms are down, you can't get through. When the arms are up, you can get through. So what happens is when a signal molecule binds with this particular receptor, that changes the shape of the gate, and it allows specific ions, such as sodium ions or calcium ions, to flow through in this channel through the receptor. So these gated ion channels, actually, even though they're fairly simple as far as not having as many steps, they are very important, particularly for the nervous system, because the electrical signals that we were talking about, some of these ligand-gated ion channels are actually activated by electricity, in addition to being activated sometimes by certain chemicals. So the electrical signals of the nervous system can cause these ion channels to open and close and allow the flow of ions in and out of the cell. Here's a good model of this. That's also on page 213 of your textbook. So here's a ligand-gated ion channel receptor, and you notice this kind of has this little closed gate portion here. The signaling molecule, the ligand, actually attaches right there. Just like we've often talked about, when that occurs, it changes the shape of the protein, and that change in shape basically opens the gate, and there's a channel that allows particular ions to pass through. And that concentration of ions, that change in concentration, causes some sort of cellular response. And then when the signal molecule leaves that particular position, the shape goes back to the way it was, and the gate closes. So basically an ion channel is a pathway for charged particles, oftentimes sodium, calcium, that are very important as far as the concentration inside or outside the cell, and they are opened and closed by certain signaling molecules. So what happens to the signal once we get inside the cell? Because there's a lot of different parts that this cell has inside of it. I mean, we have a nucleus, we have mitochondria, we have endoplasmic reticulum. So where does that signal go, or how is that signal carried once we get inside the cell? Well, that's the part that gets taken care of by what we call intracellular receptors. So we have receptors that are in the cell membrane that gather signals from the outside, but then we also have different proteins and receptors inside the cell that carry that signal to where it needs to go throughout the inside of that cell. So these intracellular receptor proteins are going to be found floating around in the cytosol, liquid part of the cell, or they're oftentimes also located in the nucleus of the particular target cell. A lot of these are basically very small hydrophobic chemical messengers. So a variety of like gases, like a nitrous oxide gas, things like that, they can easily cross membranes and activate receptors. So some of these molecules can get through the cell membrane on their own if they're small hydrophobic molecules, and then once they get inside the cell, they will activate certain intracellular receptor proteins that will start the chain inside the cell. So what we're saying is that in some cases, the first part of the signaling, the reception part, actually occurs inside the cell. But that only happens with certain molecules that can get through the plasma membrane by themselves, the cell membrane. So these would tend to be fairly small hydrophobic chemical molecules. Some good examples would be some of the various steroids and thyroid hormones of animals. They can pass through the cell membrane. Once they get inside the cell, that's when the first reception occurs, the first step occurs. One good example of some of the activity that might occur because of these particular molecules is with some of these hormone receptor molecules. They can act as what we call transcription factors. They will go into the cell, they will bind with certain proteins that will go to the nucleus of the cell and will determine which part of the genetic code to transcribe. So it will say, okay, we want this part of the genes to go to work, and that will be formed into messenger RNA, and then that messenger RNA piece will be taken out to the ribosomes to make particular proteins. So some of these chemical messengers affect what gets made inside the cell because they affect what the nucleus sends out as instructions. They affect transcription, which is the process of creating messenger RNA to send out to the ribosomes to say, okay guys, make this particular protein. And a good example would be any of the sex-related hormones, like, for example, testosterone in male animals. That's a good example. This hormone is secreted by the testes. It's circulated throughout the entire body, and it's circulated by the circulatory system to all of the cells. And it will enter all of the cells because it can pass through the cell membrane. But not every cell responds to it. Only the cells that have the receptor molecules that are inside in the target cells will bind with that particular chemical and then cause a change. That change activates certain genes that transcribe into messenger RNA, which then goes to the ribosomes and tells them build these particular proteins. So, for example, when guys go through puberty, or you're comparing men to women, and they have different characteristics, physical characteristics, well, those different physical characteristics were caused because this hormone gave the signal to the cell to build specific proteins and cause particular changes within the body. So let's take a look at a model of one of these intracellular receptors that act inside the cell, and we'll look at this hormone testosterone. So here it is floating around in the extracellular fluid. It was brought here by the circulatory system, by the blood. It's throughout the entire body. It enters through the plasma membrane. Inside there will be a particular receptor protein that binds with it. It fits into that particular slot. Then that, bound together, will enter into the nucleus. Remember, the nucleus has its own particular membrane. Once it gets inside the nucleus, it will cause the DNA to transcribe a particular set of instructions. So a portion of the DNA gets transcribed, or basically you can think of it almost kind of like written down as instructions, in messenger RNA. That messenger RNA travels out of the nucleus to all the little ribosomes. And remember, the ribosomes are kind of like the little factory workers that build the proteins. They take materials from within the cell. They use that messenger RNA as a diagram or an instruction code to build proteins. So a quick review. The first step of this three-step process of signal transduction is reception, and that is when a signal molecule binds to a receptor protein and causes a change and starts the whole process off. And the three main molecules that are involved with reception are G protein coupled receptors, receptor tyrosine kinases, and ion channel receptors. And then the last, kind of not really fourth whole category, but the last little part we talked about was the intracellular receptors, where the reception actually first takes place inside the cell instead of at the cell membrane. Let's take a break right here. We'll come back and we'll talk about the next step, which is transduction.